Hello, thank you for joining our webinar today. My name is Tara Crawford Parks, and I work as a director in our R&D department at DNA Genotek, located in Ottawa, Canada, a subsidiary of OraSure Technologies. Today, I am excited to share some insights, challenges, and solutions that are important to consider when incorporating multi-omic workflows into research studies. Including multiomics into a study workflow can provide more robust data sets that give broader insights for the system being investigated. Leveraging data from multiple omics layers can enable researchers to go beyond the genome to accelerate biological discovery and power next generation precision science and medicine initiatives. There is a growing body of evidence in the literature to support this notion, with consistent year-over-year growth in multi-omic publications as depicted in the graph here. As an example, evidence within the literature demonstrates that the salivary microtranscriptome, both host and microbial-derived RNA, can be used to distinguish between children with autism spectrum disorder versus children with typical development or developmental delays. More recently, a paper was published that identified salivary microtranscriptome targets that were differentially expressed among children with autism spectrum disorder related GI disturbances. This is exciting because this work highlights the potential for these identified targets to serve as biomarkers or therapeutic targets for future personalized diagnostic and therapeutic approaches, and is particularly significant for the multi-omic and precision medicine fields, as it highlights the strength of integrating host and microbial signatures to inform on precision health initiatives. Although these examples paint a nice picture of the value that multi-omic approaches can add to the precision medicine field, there are key considerations to keep in mind when designing multi-omic studies. My focus today will be related to some considerations specific for biological sampling methodology. Choosing an efficient and cost-effective sampling methodology becomes even more critical when incorporating multi-omics into a study. There are challenges associated with collecting multiple independent samples to capture various analytes of interest. First, this approach may increase sampling variability, which can be carried through a study workflow and can impact downstream data analysis, creating unnecessary noise in data sets. Next, collecting multiple samples creates additional logistical challenges related to sample traceability, storage space requirements, and is often associated with increased costs. Finding a solution that captures and stabilizes multiple analytes of interest in a single tube can reduce or eliminate these challenges. Finally, the sample type included in a multi-omic study can also have impact on the overall success of the study. For example, saliva has emerged as an alternative to blood with many key advantages. These include increased collection compliance due to the easy nature of collecting saliva, reduced study costs as a result of not needing a phlebotomist, simplified logistics due to streamlined storage and transport options, and using saliva as a sample type also offers an opportunity to broaden the accessibility of the study. We believe we have a solution to the sampling challenges that exist in the multi-omic space with our recently launched saliva collection and stabilization product, the OMNIgene SALIVA DNA and RNA device. With an all-in-one user-friendly design, the OMNIgene SALIVA DNA and RNA device provides a solution for intuitive saliva collection, with a wide funnel to facilitate collection and an attached cap containing the stabilizing liquid, which is sealed by a membrane to prevent spillage. Once the saliva is collected into the tube to the fill-to line, the user closes the funnel lid and the membrane is pierced, releasing the stabilizing liquid into the tube. The funnel is then removed and the tube sealed with a screw cap. Upon mixing with the stabilizing liquid, the device stabilizes DNA and RNA from multiple sources, including human, bacteria, and viruses, for storage and transport at ambient temperatures, negating the need for cold chain transport. This device tube is also barcoded to offer full sample traceability from donor to laboratory, simplifying study logistics while providing nucleic acid yields that are suitable for multiple downstream applications. This product is positioned to not only provide a solution for multi-omic studies using saliva, but has potential to power research aimed at interrogating the salivary host and microbial interface, which, as I described in my earlier example with autism spectrum disorder, has potential to accelerate precision medicine initiatives. Over the next few slides, I will describe a subset of the product development testing that was conducted and corresponding results to support this product. The OMNIgene Saliva DNA and RNA device leverages the same physical form factor of our trusted legacy device ORIGINE DX, which has been robustly validated for ease of use and collection compliance and has been on the market for over a decade. Specifically, this study evaluated the use of the product form factor across naive user populations reflective of the US general population, and were designed to evaluate real-world use of the device in at-home self-collection environments. In this particular study, 507 naive participants were recruited and provided a study kit containing the device. They were instructed to register the kit, collect a saliva sample following the instructions for use, complete a comprehension questionnaire on the instructions for use, and return the collected sample to a processing laboratory via standard postal services. Approximately 96% of participants agreed that the collection instructions were easy to find and understand in order to correctly collect their sample, while 99.5% of the donors understood how to follow shipping instructions to send the sample back to a processing lab. The OMNIgene Saliva DNA and RNA device is designed to maximize nucleic acid yields to offer experimental flexibility. Each device collects 1 milliliter of saliva, which is combined with 1 milliliter of stabilization liquid for a final sample volume of 2 milliliters, which supports up to 8 250 microliter extractions, and therefore providing ample sample to fit a diverse range of downstream assay input requirements. To evaluate the average nucleic acid yields that were obtained from a single 250 microliter aliquot, total DNA and RNA extractions were conducted to co-purify the analytes of interest for this study. DNA and RNA concentrations were subsequently determined by fluorescence-based methods, and the data demonstrate that the average nucleic acid yields per extraction aliquot were 0.90 micrograms of DNA and 0.72 micrograms of RNA. The yields evaluated in our study demonstrate sufficient quantity for input into various downstream molecular applications. Next, sample performance was tested and proven at ambient temperatures during storage and transport conditions. Here, we evaluated the stability of saliva samples obtained from the OMNIgene Saliva DNA and RNA device during room temperature storage for up to 21 days, as shown in the graph on this slide. This graphic shows the quantification cycle analysis for representative target genes across saliva samples collected in the device. Nucleic acid extractions were performed at baseline, or T0, and then again after 21 days of storage, indicated by T21. The samples were then analyzed by qPCR or RT-qPCR using primers and probes for the chosen representative gene targets. In this study, viral nucleic acid stability was evaluated by spiking samples with a representative DNA and RNA virus, while all other targets were endogenous. Each point in the graph on the slide represents an extracted sample from one individual, and the overlaying box plot represents the data median and quartiles. Although the data does show some donor-to-donor variability, which is not unexpected, the data demonstrates that all analytes were detectable following storage at room temperature for 21 days. Next, we evaluated the stability of saliva samples obtained from the OMNIgene Saliva DNA and RNA device during expected transport conditions. Similar to the previous slide, the graph here shows the quantification cycle analysis for the same representative target genes across saliva samples collected in the device. Nucleic acid extractions were performed at baseline, indicated as pre, and following 3 freeze-thaw cycles, ranging from -20 degrees Celsius or 4 degrees Fahrenheit to 50 degrees Celsius or 122 degrees Fahrenheit. The detection of each analyte was then evaluated using the same qPCR or RT-qPCR assays described in the previous slide. Similar to the room temperature stability data, this data also shows that all analytes were detectable across the donor population following exposure to expected transport conditions. Together, the data shown in the last few slides supports the use of the OMNIgene Saliva DNA and RNA device in at-home self-collection environments, while also demonstrating stability of host and microbial DNA and RNA analytes. To supplement this work, I also wanted to share a representative data set that we have generated to highlight the value that a multi-omic study can add to research initiatives. In this proof of concept study, we collected saliva samples with the OMNIgene SALIVA DNA and RNA device from 7 study participants and stored the samples at room temperature for at least 7 days. DNA and RNA were co-purified from a sample aliquot, and the quality and quantity were evaluated. The samples demonstrated high quality with the presence of high molecular weight DNA, as well as the presence of intact ribosomal RNA bands shown here. DNA and RNA yields were determined and are depicted in the graph, showing a range similar to the product development testing, which are within sufficient amounts for various downstream molecular assays. This multi-omic study evaluated metagenomic and metatranscriptomic signatures from the bacterial DNA and RNA present in the saliva samples. This figure represents the relative abundances of bacterial taxa across the 7 donors for the corresponding DNA and RNA samples. The key things that I want to highlight here are that the relative abundance of the bacterial taxa across donors varies, and that within a single donor, there are some changes in bacterial profiles between the DNA and RNA analytes. This is not unexpected when evaluating the microbiome, and further highlights the importance of collecting data from multiple analytes when designing studies to advance precision health. This study would enable researchers to understand what is present by looking at the DNA signatures and what is active by looking at the RNA signatures, allowing for broader insights from this dataset compared to just looking at DNA or RNA alone. The key takeaways that I hope I leave you with today are that the OMNIgene SALIVA DNA and RNA device provides a solution that offers scientists a simplified and streamlined saliva collection and stabilization workflow that can accelerate biological discovery and guide precision medicine strategies by incorporating multi-omics seamlessly into study workflows. With a user-friendly design that enables self-collection, sample collection can take place anytime, anywhere, and without specialized training or assistance. The device captures and preserves salivary DNA and RNA from humans, bacteria, and viruses, which adds value to researchers' needs today, while also providing experimental flexibility to expand future research. The samples collected in this device can be stored and transported with minimal logistical burden, thereby opening the door for broader collaborations and study recruitment windows that transcend geographical restrictions. In addition, we offer flexible customization services, which include design and branded materials, such as custom instructions for use with the academic institution or company logo and color palette, and other custom branded packaging options to create a solution that serves the entirety of the study workflow. You can see on the slide that there are various user touchpoints where branding can also be placed. In addition to branding, the customization options extend down to the saliva sample itself, with customized barcoding solutions, including 2D barcodes, QR codes, and accessory barcode labels to provide donor privacy while enabling traceability of the sample throughout the entire chain of custody, from shipment to a donor's home or study site and then back to the lab for processing. We hope these insights were valuable to you. As you navigate your future multi-omic studies, we take pride in our consultative approach with capabilities to tailor solutions to your individual needs. Please feel free to contact us via email for further information.